Hello, everyone, and welcome to today's live broadcast, Surveillance and AI-Powered Prediction of AMR to Inform Antibiotic Therapy. I am Sydney McNeil of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by OpGen, a precision medicine company harnessing the power of molecular diagnostics and bioinformatics to help combat life-threatening infectious diseases and fight antimicrobial resistance to improve patient care. For more information about our sponsor, please visit their site at opgen.com. Now let's get started. I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. If you have trouble seeing or hearing the presentation, report your problem by clicking on the Ask a Question box located on the far left of your screen. I would like to now introduce our presenter, Dr. Arne Materna, Managing Director and CEO of ARESgenetics, an OpGen Group company. For a complete biography on our presenter, please visit the Biography tab on the right side of your screen. Dr. Materna, you may now begin your presentation.
Hello, and thank you for tuning into this seminar. My name is Arne Materna, and I'm the CEO of ARESgenetics. And over the course of the next 30 or 40 minutes, I will speak about how machine learning, when combined with sequencing-based solutions, can help us overcome limitations in how we currently address the global challenge of antibiotic-resistant, or AMR, pathogens. But first, a few words about ARESgenetics. We are based in Vienna, Austria, and we're part of the OpGen group of companies. Together with CURATION and OpGen, we address AMR with cutting-edge molecular diagnostics and genomic testing. While CURATION and OpGen develop PCR-based IVD testing, ARESgenetics was founded to harness the potential of next-generation sequencing and AI-based solutions. I'd like to begin the seminar with a few facts on antimicrobial resistance. While the attention of many of us quite rightfully is on the impact of the current COVID-19 pandemic, another severe pandemic is on the rise. The antibiotics, which we all have come to rely upon as an effective treatment of otherwise often dangerous and maybe even deadly infections, they are slowly but steadily becoming ineffective. We see a global increase in pathogens that prove resistant against standard and even against last resort antimicrobial therapy. With already tens of millions affected by antimicrobial resistance, or AMR, and dozens of billions in euros per year in direct healthcare costs, the probably most severe consequence we face are the over 700,000 fatalities each year. That amounts to over 2,000 deaths each day. And by some estimates, these numbers are expected to rise within about a generation to 10 million per year, meaning AMR would eclipse cancer as a leading cause of death. The problem of AMR is made worse by the challenge of choosing the right treatment. Now, predicting how an infected patient will respond to antibiotic therapy is a hard problem, which is influenced by factors such as the genomics of the pathogen causing the infection. And as a consequence, only a minority of patients are receiving appropriate treatment, a fact that has become once more apparent in the overtreatment of COVID-19 patients during this pandemic with antibiotics in order to quell potential bacterial co-infections. Internal studies have shown that in fact only 15% of patients receive appropriate antibiotics, while 63% of all patients are overtreated with ineffective antibiotics. And to explain the significance of overtreatment, it is worthwhile taking a look at the emergence and evolution of AMR. As you probably know, and as every microbiologist will tell you, antibiotic compounds and resistance mechanisms evolve naturally as part of the competition between microbes, which are competing for resources in the same ecological niche. The problem arises when natural AMR mechanisms are met with inappropriate use of ineffective antibiotics, which causes any existing and newly evolved AMR mechanisms to sweep through the entire bacterial population. And whenever such a situation is then met with imperfect or poor hygiene and infection prevention and control, then outbreaks of AMR or MDR pathogens are the consequence, fueling this vicious cycle of antibiotic consumption, increasing the demand for new antibiotics, and so on. The question, therefore, is what can be done to break this vicious cycle leading to AMR? On one hand, we rely on the pharmaceutical industry to keep developing novel forms of antibiotic therapy. The challenge for pharma thereby is that the new drug development any new drug development becomes a race against the evolution of resistance mechanisms. While an encouraging number of 70 drugs is currently in different stages of clinical and preclinical development, the WHO notes in its report from this year on antibiotic drug candidates that currently over 80% of newly approved drugs can be rendered ineffective by existing resistance mechanisms. This means that drug development alone is not enough to effectively control AMR. Global health organizations are therefore calling for antimicrobial stewardship to help us regain the advantage over AMR. Antimicrobial stewardship refers to technologies and interventions that promote the optimal use and that avoid the overuse of antibiotics, which in turn not only improves patient outcomes, but also reduces the emergence of antibiotic resistance. Antibiotic stewardship, however, depends on technology for diagnostics and surveillance. While rapid diagnostic tests capable of determining whether a pathogen will respond to therapy with resistance or not have the potential to enable a more targeted antibiotic therapy, the majority of antibiotic prescriptions today are empirically informed, thanks to surveillance data. So let's take a look at the technologies that are in the clinic today. The standard of care in infectious disease diagnosis are bacterial cultures, which are routinely used for the identification of infective agents and the determination of suitable antibiotic treatment options. Whenever a culture is positive, meaning if pathogen growth is present, then so-called antibiotic susceptibility testing, or referred to as AST, is performed in order to determine which antibiotics are effective and which will likely be met with resistance. So AST determines seeks to determine the resistance phenotype or AMR phenotype. While these culture-based methods are being considered the gold standard, they are often very labor-intensive and may miss difficult to culture pathogens. The turnaround times range from 1 to 3 days or more, which is long considering that patients with acute infections require urgent care within hours. This lack of sensitivity and speed is addressed by modern molecular tests relying on PCR. Leading PCR panels, such as the tests available on the Curetis Univero system, achieve very high sensitivities and can turn around results in a matter of hours, or even pushing the limit below 1 hour these days. However, these systems can only screen for up to a few hundred biomarkers at best. This means each assay, which has to be individually developed, validated, and potentially approved by the regulators, can cover only a limited number of pathogens, and for each pathogen, again, only a very limited number of genetic markers for AMR. Given that the number of known genetic markers for AMR is today measured in the thousands, the question has to be, is there a system that could simultaneously and comprehensively screen for any pathogen and all known genetic markers for antibiotic resistance? And what if the system would not only report the detected AMR markers, but also emulate the advantages of culture by predicting whether detected pathogens are susceptible or resistant to antibiotic therapy? Here is where next generation sequencing, or abbreviated NGS, enters the picture. NGS is increasingly adopted by clinical labs, and its utility for microbial typing and epidemiology is already undisputed. NGS is therefore also in the center of attention of ARESgenetics. It is the first technology that, thanks to an enormous sampling depth, is not only very sensitive, but also captures the entire genome, allowing for an exhaustive characterization of genetic AMR markers in a single test. Moreover, such comprehensive genomic information provides a chance to use artificial intelligence and predictive modeling to emulate culture-based antimicrobial susceptibility testing and to predict which antibiotics represent suitable treatment options. With the sequencing costs as well as turnaround times rapidly decreasing, trying to overcome the shortcomings in the current standard of care through sequencing-based predictive antimicrobial susceptibility testing has firmly emerged as a focus for our development teams. As we set out to develop rapid and accurate sequencing-based methods for the comprehensive detection of pathogens in AMR and the prediction of bacterial response to antibiotic therapy, we quickly realized that a key component in the entire workflow were AMR marker databases. When we evaluated several public AMR marker databases for their suitability for predictive AST or resistance prediction, we recognized several shortcomings that I'd like to present to you here. One issue was that genetic AMR markers in public databases were annotated at the level of antibiotic classes, which was too coarse for machine learning, considering that different antibiotic compounds in the same class can be affected differently by any given AMR marker or mechanism, for example, due to differences in enzymatic affinities. Therefore, raising the resolution of marker annotation in the databases from antibiotic class to the compound level substantially improved predictive modeling. Another issue we have identified was that simple rule-based models for individual markers tended to overgeneralize because multiple genetic factors can, for example, synergistically or antagonistically contribute to the AMR phenotype. We needed to adjust for biology and allow for polyvariate predictive models. Based on these and other findings, we decided to develop ARESdb, a comprehensive AMR knowledge base designed from ground up to enable predictive AST. ARESdb today is a globally leading database of curated AMR markers and information about resistance derived from genotypic and phenotypic data. What sets ARESdb apart from other databases is not only comprehensiveness, but also that it harbors a continuously growing collection of clinical pathogen genomes and associated validated AST measurements that determine how a given pathogen in this database has responded phenotypically in the lab when exposed to a wide range of antibiotics. This additional data layer offers two unique benefits. The genotype-phenotype data association can be used for machine learning, specifically in order to discover novel biomarkers for AMR, and on the other hand, to train predictive models that can predict how pathogens respond to therapy based on genomic data, a process we refer to as predictive AST or PAST because it generates, just like conventional AST, a antibiogram. Currently, the models to predict antibiotic susceptibility are trained as stacked ensemble models, which associate phenotypic AST results and a variety of genetic markers, including, for instance, KMERs, gene models, mutation score annotations, and so on, within the ARESdb. This model-based approach can be complemented by a rule-based approach, taking into account the interplay between multiple resistance mechanisms and antibiotic compounds. As the figure in the middle illustrates, we're not only relying on the presence-absence of certain well-known resistance genes, but we also consider changes in non-coding regions of the genome, as well as novel markers, for example, in regulatory regions and other parts of the genome that our predictive algorithms have discovered. We demonstrated in several studies the high categorical agreement between the standard of care results with the resistance phenotypes we predicted based on genomic data. These studies, for instance, also include the first and only multi-center evaluation, which successfully demonstrated the feasibility and the performance of predictive AST for 13 pathogens causing lower respiratory infections. As you can see in this graphic on the right side, we obtained a cohort of patients for which culture positive isolates were subjected to standard of care testing. more specifically to AST using both microdilution testing and MALDI-TOF for pathogen identification. In parallel, we extracted DNA from such isolates and subjected them to genome sequencing and downstream bioinformatics analysis using AREScloud. The results were compared for concordance. This slide is quite busy, but allow me to walk you through some of the results shown here. Along the horizontal axis, you can see the 13 pathogens that were identified by the standard of care. Along the vertical axis, you can see the different antibiotic compounds grouped into compound classes, and the intersections in this matrix correspond to the % categorical agreement of our resistance prediction with the measured phenotype in the laboratory. If you now exclude those pathogens which are intrinsically resistant against certain compounds, here shown in blue, and those in gray for which the standard of care did not yield any result, then the white intersections are the ones to focus on when comparing ARES-PASD with culture-based antimicrobial susceptibility testing. For the majority of these intersections, now highlighted in green, we were able to demonstrate high concordance, reaching for most of the pathogen drug combinations values significantly above the regulatory threshold for ASD devices, which is at 90%, often achieving values up to 100%. Studies like this are not only important for clinical validation purposes. We also embrace them as they give our data science teams clear indicators on which pathogen drug combinations to focus our efforts on for predictive model development, as well as for database curation. Here, the result of such an effort to improve model performance for a given drug pathogen combination through focused biocuration and model training. Together with partners such as reference labs, we generate and aggregate isolate genomes and the corresponding phenotypic data on AMR. The data here show that by increasing the number of specific isolates, as you can see here along the x-axis, not only the categorical agreement, but also the so-called very major error rate, which is the false susceptible rate, shown here on the y-axis, can be systematically improved and reduced here to levels that are in line with IVD device regulation. The benefit for our partners thereby resides in being able to accurately predict treatment response directly from genomic surveillance data, and of course, in having access to an up-to-date AMR marker database for their surveillance efforts. Over the next couple of slides, I would like to talk about the development of solutions based on ARESdb and predictive modeling and the validation of such solutions. Here, a number of products and services that are essentially enabled by ARESdb. Starting with testing as a service, which is offered through our Vienna-based next-generation sequencing services lab, we are offering ARESiss. ISS stands for isolate sequencing, for example, for infection prevention and control, surveillance, and clinical R&D. In contrast to ARESiss, ARESupa is a universal pathogenome assay which seeks to identify pathogens and AMR in native specimen and without the need for prior culture. Both assays accept either DNA. ARESiss also accepts isolate material, and ARESupa accepts native patient or environmental specimen. It's important to point out that ARESupa at the moment is in early access testing with customers in different countries. In addition to testing as a service, ARES also offers interpretation as a service through a bioinformatics product called AREScloud. The AREScloud is a bioinformatics cloud portal for pathogen identification, typing, as well as the AMR characterization. It is powered by our AI-based predictive algorithms and the proprietary ARESdb. Here, another visualization of ARESdb and enabled solutions. I'd like to start by providing further details on ARESiss. Before I continue, I'd like to apologize in case there is acoustic feedback. I'm getting some feedback every time I switch the slides, and I hope it does not affect this seminar. Back to ARESiss, the isolate sequencing service. As I mentioned, the service is offered through our Vienna-based service laboratory, and the service is designed to meet the requirements of clinical microbiologists. We accept either culture or DNA as starting material, can deliver short, long reads, and rapid turnaround times are available upon request. Clients can conveniently access raw data, bioinformatic analysis results, and the final reports through the AREScloud portal. The reports identify primary and secondary pathogens, as well as MLSD and core genome MLSD genotyping results. In addition to antibiogram prediction, we offer a comprehensive characterization of detected markers for AMR, plasmids, as well as virulence factors. We also deliver information on strain relatedness and conduct detailed outbreak analysis, and we can offer custom bioinformatics support. To ensure the accurate performance of our platform, we routinely validate our workflows and publish the results along with the validation protocols. Here are the example of a study validating ARESiss and the analysis following CLIA guidelines. CLIA is a US standard for laboratory developed testing. The performance was assessed recording a number of parameters, including accuracy, repeatability and reproducibility, as well as sensitivity and specificity. The study showed that ARESiss proves 100% accurate and reproducible for pathogen identification and genotyping. Also, AMR marker detection was highly accurate with sensitivities and specificities exceeding 95 and 99% respectively. With nanopore sequencing becoming increasingly available, we have just conducted a multi-center validation study of ARES isolate sequencing using the ONT platform. ONT hereby stands for the platform by Oxford Nanopore Technologies. Nanopore sequencing is an interesting platform for clinical microbiology for a number of reasons. For one, the turnaround time is low, theoretically enabling same-day reporting and workflows as short as 4 to 6 hours. The technology also generates long reads, which offer interesting advantages for the assembly of bacterial genomes or for the recovery of plasmids. Finally, the small footprint and the low capital equipment cost, as well as the per-base cost, make this an interesting platform for clinical or surveillance applications. Nanopore sequencing works by translocating a single DNA strand across a biological pore and a membrane, or through a biological pore and across a membrane. A steady flow of ions through that pore creates thereby a current which can be measured and which fluctuates in a DNA sequence-specific pattern as DNA is translocated through this pore. The downside is that both the nanopore itself as well as the DNA translocating effect rely on live biological proteins, which introduce biological variability into the system, leading to less accurate base calling compared, for instance, to Illumina short reads. In order to determine if and how less accurate long reads affect the bioinformatics we use for ARES isolate sequencing, we conducted a multi-center validation. The well-characterized set of reference materials was subjected to DNA extraction by using kits by different vendors, and the resulting DNA extracts were aliquoted and sent to ONT-enabled partner labs, so laboratories with access to Oxford Nanopore technology platforms for library preparation and sequencing using the 10.4 flow cells and the rapid library preparation chemistry by ONT. Indeed, the base calling quality was reduced. We obtained quality or Q scores on the Phred scale of 11 to 12 for ONT compared to much higher Q scores in the range of 30 we typically see with Illumina data. However, the assembly process that follows resolves some of these problems. After de novo genome assembly, we assess the average nucleotide identity of the assembled genomes with the known public reference and determined that the error rate was only 3 to 4-fold higher compared to Illumina-based assemblies. Despite this remaining unresolved difference in sequencing quality, the ARES bioinformatics pipeline for ARES isolate sequencing performed very well. The accuracy and reproducibility for pathogen identification was robustly 100%. We also obtained high accuracies for AMR marker profiling and plasmid detection, which were only slightly below the performance of Illumina data. Most satisfying was the fact that the antibiogram prediction using predictive ASD performed very well, with an accuracy of up to 97% and in line with Illumina data. We believe that this performance is owed to the stacked ensemble modeling approach I described earlier. This approach associates not just one, but a number of different genomic features with phenotypic resistance data, and therefore it appears to be rather robust to NGS error. The only area where nanopore technology can further improve is bacterial typing, which at the moment appears to be more sensitive to sequencing error. But we have enrolled with Oxford Nanopore Technologies in their Q20 program, promising drastic quality increases, and are preparing the next evaluation as we speak. I'd like to shift gears and talk a bit about the software platform AREScloud, which we use to evaluate data from ARES isolate sequencing. AREScloud is a user-friendly cloud-based portal which fully automates the analysis of long and short read data from, for instance, clinical isolates. AREScloud returns comprehensive reports with information on sequencing quality and contamination, pathogen identity, and genotype. Also included are predicted antibiograms, which clearly identify implicated antibiotics. From here, users can drill into rich information on individually detected AMR markers, their prevalence, and prognostic performance. Also included is information on plasmids and virulence factors. All reports can be explored online within the AREScloud user interface or exported with the click of a button. For convenience, the AREScloud also includes a set of comparative analysis tools for the investigation of local bacterial outbreaks. I would like to conclude the overview over ARES products and services with the ARESupa. This universal pathogenome assay is designed to detect pathogens and AMR markers directly in native and potentially polymicrobial patient specimen or environmental samples. The culture-free detection of AMR markers in native materials is quite challenging, even for ultra-deep shotgun metagenomic sequencing, as I will show you in a minute. The issue is that accurately and reproducibly recovering individual AMR markers, such as, for instance, a mutation in a gyrase gene, is very problematic, as the majority of DNA in such samples comes from human or environmental background. To overcome what essentially is a sampling problem, we have therefore developed an assay relying on target capture sequencing. Thanks to ARESdb, we are able to design baits that comprehensively cover known AMR markers, including resistance genes, as well as resistance-inducing mutations. The 8,090 baits capture over 9,000 AMR genes and alleles, and we ensure that, unlike for metagenomic sequencing, near-complete marker coverage of close to 100% and twofold tiling of each AMR locus is accomplished. This slide shows the performance of metagenomics for AMR marker detection in 4 environmental samples. As you can see in the table on the left-hand side, the number of AMR markers that can be recovered is rather small. And as the chart on the right-hand side shows, the AMR marker coverage is in fact sequencing depth dependent, which is further impacting the cost and the sensitivity of deep shotgun metagenomic sequencing. Now let's take a look at how ARESupa compares. As you see, the number of AMR markers which can be reproducibly recovered drastically increases compared to metagenomics. And also the marker coverage is consistently near 100% and independent of next generation sequencing depth. So in summary, the ARESupa represents a sensitive and cost-effective alternative to metagenomics. It also offers, thanks to ARESdb informed design, an AMR marker panel which in terms of its comprehensiveness eclipses with more than 9,000 covered markers any other AMR panel we are aware of. Shifting gears once again, I will now present a number of clinical cases of newly emerging antimicrobial resistance and the attempt by researchers and clinicians at the Johns Hopkins University and Hospital to define the associated AMR markers and mechanisms. The clinical cases focus on resistance to cefiderocol, which makes them particularly concerning because cefiderocol is a relatively new compound reserved for last resort therapy. A few words on the background. The magnitude of carbapenem resistance in gram-negative bacteria has reached an alarming level. In recent years, several new antibiotics have become available for clinical use. However, these new agents have a limited spectrum of activity when compared with cefiderocol. Cefiderocol has been developed following an interesting approach, and it is, to my knowledge, still the first and only compound of this type that has passed regulatory approval. Cefiderocol is a synthetic conjugate composed of different moieties that promote, for instance, entry into the bacterial cell and antibacterial activity. There's also moieties that improve its stability to beta-lactamases. For example, a cephalosporin moiety inhibits cell wall synthesis and acts in an antimicrobial fashion, and a catechol type siderophore moiety gains entry into the bacterial cells by using active iron transporters, making the drug essentially independent of usual points of entry, such as porine channels or efflux pumps. Cefiderocol is an intriguing compound with great promise for clinical efficacy against a broad range of aerobic gram-negative pathogens. And as a consequence, it is important to be able to diagnose and understand resistance mechanisms and avoid inappropriate use of this antibiotic. In a first study published in Microbial Drug Resistance this year, the authors sought to identify mechanisms leading to baseline resistance against cefiderocol. Surveillance data empirically show that the compound is 100% active in Enterobacteriales, which have not previously been exposed to cefiderocol. However, carbapenem-resistant Enterobacteriales, or CREs, exhibit an increased minimal inhibitory concentration at which cefiderocol is effective, even if they have not been predisposed to cefiderocol. In an attempt to identify baseline mechanisms, the authors collected and sequenced 56 CRE isolates from 8 different species, predominantly Klebsiella pneumoniae, E. coli, and Enterobacter cloacae complex. The sequencing results were analyzed and queried against the ARESdb using the AREScloud pipeline. The results indicated that cefiderocol resistance in CRE likely has no single mechanism leading to baseline resistance. Instead, cefiderocol resistance seems to result from a combination of heterogeneous mechanisms, including different beta-lactamases and AMC beta-lactamases. Approaching the problem from a different angle, the team expanded their view to include Pseudomonas aeruginosa with a difficult-to-treat resistance phenotype, or DTR phenotype. These are all DTR Pseudomonas aeruginosa isolates are resistant to essentially all beta-lactams, fluoroquinolones, and therefore pose a significant clinical challenge. DTR Pseudomonas aeruginosa strains even respond with elevated resistance to new drug combinations like ceftolozane, tazobactam, and others after being exposed to these compounds, which leaves cefiderocol in these cases as a precious last alternative. After reports that DTR Pseudomonas aeruginosa strains also develop cefiderocol resistance after treatment with ceftolozane and tazobactam, the team collected 16 pairs of isolates before and after exposure. The isolates were sequenced and the genome analysis was conducted using the AREScloud pipeline. Once again, a diverse set of resistance mechanisms was identified. And again, the AMC beta-lactamases were among them. This time, structural mutations more clearly implicated the AMC omega loop. In the third study, most recently published in Clinical Infectious Diseases, the team studies a case of progressively evolving cefiderocol resistance, discovered in a patient suffering from lymphoblastic leukemia. Sequencing identified an E. coli strain, which was producing the metallobeta-lactamase BLA-NDM5. make this work. Here we go. 5 isolates were sequentially collected during the treatment and subjected to RNA-seq for gene expression analysis, to phenotypic characterization, and to whole genome sequencing and AREScloud analysis. The expression analysis data shown here further implicated BLA-NDM5 and showed that an increase in copy numbers and an elevated gene expression levels correlated with increasing levels of resistance to cefiderocol. The phenotypic characterization confirmed that differential gene expression of BLA-NDM5 is in fact associated with cefiderocol resistance. Resistance to cefiderocol can again be reduced, or if you will, susceptibility to cefiderocol increased, when inhibiting BLA-NDM5 through EDTA-mediated binding of its cofactor zinc, as shown here in the disk diffusion assays. Finally, genome sequencing and AREScloud analysis of all isolates was conducted. The data showed that BLA-NDM5 was residing in a mobile element, and it also revealed mutations which implicated 3 other genes listed here. I would like to focus on 2 genes in particular, ENVZ and MAR-R, as both genes are involved in modulating gene expression. ENVZ has been associated in the literature with an increase in resistance to cefiderocol. It also encodes for a cell signaling protein controlling the DNA binding transcriptional regulator AMR. Remarkably, MAR-R is, as a DNA binding repressor, also involved in transcriptional regulation. In fact, it is involved in the global regulation of over 80 genes in E. coli. Also interesting is the fact that MAR-R has been associated with elevated resistance to antibiotics through its contribution to the acidification of the cytoplasm, potentially supporting hydrolysis. Of course, further work is required to elucidate the interplay between BLA-NDM5 and mutations in transcriptional regulators in causing resistance to cefiderocol. These studies, however, show that whole genome sequencing and AMR marker discovery is important to identify biomarkers not only for the presence, but also for the upregulation of AMR-inducing factors such as BLA-NDM5. With that, I will conclude the seminar and thank our collaborators at Johns Hopkins University and Hospital, Dr. Pranita Tama, Dr. Patricia Simner, and Dr. Judith Bergman. The authors will present more on their research into detecting and treating AMR in an upcoming webinar, and we will of course share the dates and registration details with you in the follow-up. So stay tuned. Of course, I would also like to thank you for your attention, and now use the time and invite your questions.
Thank you, Dr. Materna, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, we are a reference laboratory and currently have to independently generate genome data and antibiograms for each isolate. Can Ares technology be used to combine both steps into a single workflow?
Thank you for that question. Can you in fact hear me? Does this work still?
Yes, we can hear you.
Excellent. All right, perfect. Well, the answer to this question is yes. In fact, quite a number of our partners are exploring the opportunity to gain deeper insights into phenotypic resistance directly from isolate genome data. And their main motivation is to save time and cost by avoiding conventional AST, especially in cases that are time sensitive. Examples here include, for instance, rapid or positive blood cultures. We have developed predictive models for over 100 antibiotic compounds and are continuously expanding the ARES databases with a focus on escape pathogens and healthcare associated pathogens. If you have any specific questions, for instance, with regards to supported pathogen drug combinations, please don't hesitate to let us know either here or by getting in touch. I hope that answered that question.
Wonderful, thank you. Our next question is, AMR profiling and predictive AST, especially in samples we collect for infection prevention, is very interesting and would likely help us to improve empirical selection of antibiotic treatments. How can our microbiology lab access the ARESdb?
Okay, that's another good question. We have in fact built a user-friendly web portal that I also referenced in the presentation called AREScloud. AREScloud is not only used by our own NGS service lab in Vienna to deliver results to our customers, but it enables labs with their own NGS capacity to run queries against the ARESdb. AREScloud essentially automates the entire bioinformatic analysis workflow and returns comprehensive information on AMR, detected genetic markers, and their prognostic performance. All of this is delivered on top of the reports you'd conventionally expect for isolate genome data, including information on sequencing quality, pathogen identity, MLSD and core genome MLSD typing results, plasmids, and so on. And AREScloud can be easily accessed through our website, www.ares-genetics.com.
Perfect, thank you. It looks like we have one more question. We are excited to hear that ARESgenetics is publishing validation protocols for the isolate sequencing workflow to support other labs in their adoption of NGS. We are planning to establish NGS capabilities in our lab next year. Do you offer some kind of service that would help labs with the setup of this or similar workflows?
That's an interesting question. We're happy to share studies, protocols, reference materials, and of course, we're happy to elaborate on them. However, to be compliant with regulations, I need to point out that the actual validation of laboratory developed tests has to be independently carried out by any lab that is essentially seeking to implement these LDTs into their lab environment. But lightening that burden for labs with access to NGS and AREScloud and enabling them to generate pathogen data with essentially the same accuracy as our Vienna-based NGS lab, that was in fact one of the main motivations for us and has remained one of the main motivations for us to publish and share the validation results, protocols, and so on for the assays that we develop at ARESgenetics.
Perfect, thank you.
I can see there's another question that has come in just now. Can you see it as well?
Yes, I can. Our next question is, what is the accuracy of predictive AST? Was it correlated with the AST done at labs?
Yeah, this is a very good question. And sorry if that wasn't clear enough in the presentation. This is in fact correct. The accuracy of predictive AST is measured in terms of agreement with phenotypic traditional AST conducted under controlled and standardized conditions in clinical labs. So this is how we determine the accuracy. Also, the study I was referencing in which we performed a multi-center evaluation of predictive AST is comparing the results of the prediction with the phenotypic results conducted, as I said, under controlled conditions in a lab. So it's very much a comparison of in silico generated results with lab results. And in this case, we give the lab results the benefit of the doubt and are assuming that they are the ground truth for the purpose of this evaluation.
Perfect, thank you. Thank you again, Dr. Materna, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. We would like to once again thank Dr. Materna for his time today and his important research. We would also like to thank LabRoots and our sponsor OpGen for underwriting today's educational webcast. You can view the webinar on demand. LabRoots will alert you via email when it's available for replay. That's all for now. Until next time, goodbye.